The growth and reproduction in bacteria. The bacteria also reproduce in both types, sexually and asexually. The first of all, the method, this is a very simple method that is known as the binary fission. This is asexual method for the reproduction of the bacterium. The bacterial growth refers commonly to increase in number of bacterial cells. Bacteria increase in number by an asexual means of reproduction that is known as the binary fission. In binary fission, the parent cells enlarges, its chromosomes duplicates, and plasma membrane pinches inward at the center of the cells. In this process, the one cell of the bacterium divides into two cells by multiplying the nucleoid as well as the plasmid. Similarly, the both type of the structures divide internally. Externally, the plasma membrane is also divided, pinches in this manner, and form the two cells from the one cells. This mechanism is usually observed in the bacterium that is known as the binary fission. When nuclear material has been eventually distributed, the cell wall grows inward to separate cell into two. This sequence is repeated at intervals by each new daughter cells, which in turn increases the population of the cells. Once the division is complete, bacteria grow and develop their unique features. The interval of the time until the complete or the completion of next division is known as the generation time. That is, generation time means that the interval or the time that is between the one division of the bacterium and the next multiplication or the reproductive division, between the time between two reproductive cycles is known as the generation time. The growth of the bacterium is generally recognized on the basis of their growth periods. This has been divided into four phases. Number one, the lag phase. It is phase of no growth. Bacteria prepare themselves to division. In these conditions, bacteria prepare themselves to prepare for the division. In the second phase is the log phase. It is the phase of the rapid growth. Bacteria divide at exponential rate. In this, at these conditions, the bacterial growth is maximum, and then at the third phase, that is known as the stationary phase. The stationary means solitary, और it is उर्दू में कह सकते हैं इसको साकन, यानी कि ये मुतहर्रक नहीं होता, रुक जाना. The bacterial death rate is equal to the bacterial rate of the reproduction and multiplication. In these conditions, the number of the cells of the bacterium remains constant. The third is the death phase. Bacteria start dying. Here the death rate is more than the reproduction rate. The death rate more than the reproduction rate started over here. When we draw a line from here towards the right side, and this line is consisted of the from upper of zero is upper as compared to this. However, it means that the death phase is initiated from here and stopped or the end from here. It means that the death phase, the number of the death phase, the number of the bacterial cells is greater as compared to their reproductive cells. It means as such or the as many as the number of bacterium as produced during the growth phase, all these bacteria died at the end phase, that is known as the death phase. Bacteria lack traditional sexual reproduction and mitosis. However, some bacteria transfer genetic material from a donor bacterium to a recipient during a process that is known as the conjugation. Some conjugating bacteria use specialized sex pili to transfer genetic material that, that is known as the conjugation produces new genetic combinations that may allow the resulting bacterium to survive under the great variety of the conditions. So conjugation is the only process from where the bacterium transforms their genetic material from one cell or one bacterium to the other bacterium. In this process, the reshuffling of the genetic information is carried out.